Immunological screening. Immunological screening, it is widely used for the screening or the detection of expressed product. Immunological screening, it involves the use of specific antibodies against the target protein. I think you have the idea that antibodies, they are highly specific in their reaction, and specific antibodies, they can react against specific determinants on the target protein. And one of the advantage is that if the antibodies, they are available for different expressed proteins, so they can be used against such targets. So this is one of the advantage of immunological screening, that the different antibodies, they can recognize different set of proteins. Now ये जो antibodies हैं, ये target protein पे एक खास किस्म के एंटीजेनिक डिटर्मिनेंट्स के साथ रिएक्शन करती हैं, और ये जो डिटर्मिनेंट्स हैं, इनको कॉमनली हम एपिटोप भी कहते हैं. नो व्हाट आर एपिटोप्स? Epitopes, they are specific amino acid sequences that can fold in a proper three-dimensional conformation from rest of the protein. And such specific sequence of amino acids, they are called as epitope or simply antigenic determinants. And these antigenic determinants, they are produced in different type of proteins in different conditions. Like if protein, it is expressed incompletely, even then epitopes are formed on such incomplete proteins. And even the protein, if they are expressed in hybrid form or chimeric form, which is called as the fusion proteins. Fusion proteins, they are the product of two genes, and that's why they are called as the fusion proteins. And fusion proteins also have such epitope or antigenic determinant. Under some circumstances, if host is subjected to high temperature, and under these conditions, the target proteins, if they are denatured, so even denatured proteins, they also maintain the epitope. So this method has the advantage that antibodies, they can react with such determinants or epitopes on target proteins and make the detection of the expressed product easy. So immunological techniques were used in late 1970s to detect expressed product, and this method mainly first time was used by some scientists like Broom and Gilbert in 1978. So they explained the immunological screening of the expressed protein product. Their strategy was based on the fact that target proteins, they can bind with some plastic materials like the polyvinyl, and after polyvinyl binding, then such polyvinyl sheets can be further used to detect the target protein by using another antibiotic. Now practically, what we can do during the screening of proteins by using immunological techniques. So here, the recombinant colonies that express our target protein, they may be grown on a suitable agar medium where they will express its gene product, and after expression, the cells can be lysed by using different chemicals like chloroform can be used, and even the recombinant colonies can be sprayed with virulent phages so that plaque formation may be there. So in any case, cell lysis is there that may be mediated by chloroform or phage particles. And when lysis is there, then expressed proteins, it can be shifted to the polyvinyl sheet. So what happens in this case? The replica plating, it is required. तो हमारे पास एक कॉलोनीज का मास्टर सेट होना चाहिए जहाँ से हम अपने क्लोन्स को रिकवर कर सकें. तो कॉलोनीज के लाइसिस के बाद हम क्या करते हैं कि पॉलीविनाइल शीट जिस पे हमारी स्पेसिफिक एंटीबॉडी है अगेंस्ट स्पेसिफिक एंटीजेनिक डिटर्मिनेंट, उसको हम कॉलोनीज की सरफेस पे रखते हैं, एगर प्लेट पे रखते हैं और उसको कुछ देर के बाद हम उठा लेते हैं। और उसके बाद हम जो लेबल्ड एंटीबॉडीज हैं आर रेडियो लेबल्ड एंटीबॉडीज दैट मे बी iodine 1 to 5 labeled IgG antibodies. They can be used to detect the target antigen, and after that we can detect the radioactivity by X-ray film. So in this way, this technique, by using two antibodies, which is a sandwich technique, where the antigen it is present between two antibodies. First, that is binded with polyvinyl sheet, and then antigen is there, and then antigen is detected with radio labeled IgG antibodies, and these secondary antibodies, they are labeled with radioactive iodine. So what is the basic outline of immunological screening? We are going to explain with the help of diagram. As I told you that polyvinyl sheet it can be used, and this polyvinyl sheet it is having affinity for IgG antibodies here. So IgG antibodies, it is bounded with polyvinyl sheet, and after the cell lysis, this polyvinyl sheet along with unlabeled IgG molecules, it is placed on the lysed colonies. If the antigen it is expressed, antigen mean if our target protein it is expressed with some specific antigenic determinants, then IgG antibodies will react it. Then the sheet can be lifted from the colonies, and then it is subjected to second antibody reaction. Now the second antibody, which is again IgG antibodies, it is labeled with iodine 1 to 5, so it is radio labeled, and then this radioactivity it can be detected with X-ray film. So this is the basic immunochemical detection that was used by Broom and Gilbert in 1978. In immunological screening, basically the method is to use a sandwich technique, where first antibody it is used that is bounded with a polyvinyl sheet, and then the secondary antibody, which is radio labeled, it is being used. So for sandwich technique, no, actually the target protein must contain two antigenic determinants, one for the first antibody and then for the radio labeled antibody. And even if the fusion proteins are there, then the two proteins must contain antigenic determinants or epitopes, so that their screening may be possible. So this is the basic technique where we can screen or detect the target protein by using immunological screening.